Welcome everybody to today's Wednesday seminar. So I'd first like to acknowledge that we're meeting on the lands of the Wurundjeri people of the Kulin Nation and pay my respects to their elders, past and present, and their emerging leaders, and acknowledge that the Aboriginal cultures are the longest living culture in the world, and they're the first scientists, and we can learn an awful lot from the way from what they have learned about biological systems and many others. So today it is a great pleasure to welcome Professor Joseph Takahashi here to give our Wednesday seminar. He's come all the way from Texas, and we had him at Lawn Genome for a few days already, so we've been really fortunate to have him here. And I first became aware of Joseph's work because he was doing some really cool ENU mutagenesis screens when I was doing ENU mutagenesis screens, and I loved that he got to name the factor that he found that is the master regulator of circadian rhythm. He got to name it, and it's called clock. Which is of course great, and I'm sure he's going to tell you an awful lot more about that. But the ingenuity of those screens and how he's been able to create a whole field of understanding the molecular regulation of circadian rhythm is really quite inspirational. So I'm going to leave most of the time to Joseph so he can tell you about his latest work, because we're really looking forward to your talk today. Thanks, Joseph.
Thanks, Marnie. It's really a pleasure to be here. Thank you for inviting me. So as we all know, we live on a planet that has a 24-hour day-night cycle. One way to think about that environmental cycle is it's really an energy cycle. If you were a plant, that would be obvious to you. You'd photosynthesize in the daytime, and you might fix nitrogen at night. But even mammals have an energy cycle in all of our cells every day. And I'll tell you more about that. But throughout evolution, essentially all living systems have evolved internal timing systems to anticipate the Earth's rotation. And the conserved motif for all of these clocks that have evolved is a transcriptional negative feedback loop, which takes about 24 hours to complete. In different forms of life, the specific gene sets are different. So perhaps clocks evolved independently multiple times. But in animals, they're highly conserved across all species. And so I like to point to this paper as really the beginnings of our molecular understanding of the circadian clock system. So this is a paper published by Ron Knopf and Seymour Benzer in 1971. Ron was a student with Seymour. This picture here is taken much later in 2000. And what Ron did is he did an EMS screen in Drosophila for circadian rhythms. His screen was eclosion time. Drosophila closed at dawn, which is clock control. And he just looked for colonies of flies that had closed too early or too late or didn't have a rhythm. And incredibly, he found 3 mutants. So what's shown below, I'm not sure the best way to point because I don't have it here. Oh, it does work. Okay, so here, these are activity records of individual flies that he did later from these mutant lines. This is the wild type stock. It has about a 24-hour period. He found an arrhythmic mutant, has no circadian rhythm. And then he found a short period mutant that's about 18 or 20 hours in period. And he found a long period mutant that had a 28-hour clock. Now this is an X chromosome screen. So all 3 of these mutants map to a single locus, which he named the period locus. And many years later, in 1984 and 85, 2 labs, Jeff Hall and Michael Rosbach Brandeis and Michael Young at Rockefeller, eventually cloned the period gene. And at that time, the predicted amino acid sequence that occurred was not revealing at all. We could not really tell what the protein was going to do based on its amino acid sequence. And those 2 labs actually went down a number of different blind alleys that were incorrect at the time. But eventually, they got it right and figured out that period is a transcriptional regulator in 1990 or so, the Brandeis group. And of course, in 2017, those three individuals were awarded the Nobel Prize for the cloning of PER and describing how it worked. Now, since 1984, 85 or so, all of us working in circadian rhythms who are interested in molecular biology and biochemistry tried to clone period homologs in mouse and human, and we all failed. And that's because it's interesting. At the nucleotide sequence level, you can barely recognize the gene between, say, a fly and a human genomic sequence. It's not conserved at all. It's not until you translate it into the predicted amino acid sequence that you can actually recognize it. So because of this, we knew that genes were important for circadian rhythms in a general sense. And we had the sense that they were going to be different from known pathways that were already being studied by different biologists. And so what we decided to do was to take a step backwards and go back and do a forward genetic screen, similar to what Ron and Seymour did. And so very talented postdoc in my lab, Martha Vitaturna, undertook this screen in early 1990 or so. And we learned at the time, we weren't geneticists back then, that you could do high efficiency immunogenesis with ENU in the mouse. And we collaborated with Bill Dove, who's at the University of Wisconsin-Madison, who was doing a dominant screen for an enzyme in the liver. So he was immunogizing mice, and we got together with Bill. He screened the mice and then sent all the negatives to Evanston, where we were. And then Martha screened them at the behavioral level. And so what we use is the spontaneous locomotor activity rhythm of the mouse, which you can measure by putting the wheel in a cage. You have a computer that continuously monitors the wheel, and then we plot the data as a raster plot, which we call an actogram. And this is a double plot. So each line is 48 hours long in this representation. That's just so you can see the pattern more clearly when it crosses over midnight. Okay, so at the beginning of this experiment, which is 50 plus days long, the mouse is on a light cycle, indicated at the top with the green bars, and the mouse runs every night, okay, here. And then on this day, we transfer the mouse to continuous darkness so that the mouse no longer has a synchronizing light cycle, and then it will exhibit its own endogenous periodicity. And so what you can see is the mouse is, this is real-time clock time on the x-axis. So the mouse is waking up a little earlier each day, okay? So the activity band drifts to the left. And that means the period is a little shorter than 24 hours because it's waking up earlier each day. This is typical for C57 Black 6J mice. They have a period that's average of 23.7 hours with a very tight distribution. And so in this screen, which is a dominant screen, we found a mouse that had a 1 hour longer period. It was about a 25-hour mouse. And it was a heterozygous mutant. And this record here shows you the grandchild of that founder mutant mouse after producing additional heterozygotes and intercrossing them. And so in the first litter that we looked at, there were only 4 pups. 1 was wild type, 2 were heterozygotes, and 1 was homozygous mutant, this mouse right here. So it was a perfect Mendelian ratio. So this mouse is really interesting because it has a 28-hour period. It wakes up 4 hours later each day here in darkness. It can still synchronize to a light cycle, but here I think you can see it's waking up much later each cycle. And then after about 2 weeks, the circadian rhythm damps out, and what's left is this episodic high frequency rhythm, which is not controlled by the circadian clock system, called an ultradian pattern. It's controlled by a completely different system we know now. So this mouse, we could show the mutation was caused by a single gene. We mapped it to mouse chromosome 5. The mutation was semi-dominant. And from that point on, this is in the mid-90s or so, we then went on to try to clone the gene. And so back then you had to use positional cloning. We had to find map mutation and high resolution. We collected 2,600 meiotic events to find map the locus. Once we got to that region, no one had been there before, hadn't been cloned. Genes had not been identified, so we had to physically map the region. We did that with bacterial artificial chromosomes and yeast artificial chromosome clones. And then to find the gene, we had to use all these various techniques that nobody's heard of today, except for shotgun sequencing, which worked in the end. We just brute force sequenced that back contig to identify the genes. And eventually we were able to show that there was a transcription unit that was about 100 kilobases in size. It had 24 exons, one for every hour of the day. And the predicted amino acid sequence of that gene is shown here. It was a BHLH PAS AB transcription factor, we now know. So going from that mouse to the gene, led by these two individuals, David King, graduate student, and Marina Antok, a postdoc in my lab, took 30 person years of effort between 1994 and 1997. So 10 people in my lab worked together as a team for 3 years to do all the different things we had to do to identify the gene, or find map it, identify the gene, clone the region, and then prove this was the gene by transgenic rescue of the mutant phenotype in mice. That was done by Marina. So this, at the time, this is 1997, there was the period gene from flies. Then there was a second gene discovered 1995 called timeless that was a partner of period. And then clock was actually the third gene in an animal to be discovered. We then used clock in a yeast-2 hybrid screen to find its potential partners. And in that screen, we identified another BHLH PAS protein called BMAL1, which is shown here. It's also, you can see in this diagram, also BHLH PAS AB. We solved the crystal structure of the BHLH PAS AB domain about a decade ago at Southwestern. And this turns out, by dumb luck, to be a master transcriptional regulator of the clock system. I say dumb luck because it was the first gene first mutant that we isolated. We've isolated many more mutants subsequently. We can talk about that afterwards in our workshop. And had we found any of those first, we'd be stuck with no information because no one knew the function of those genes back in the 90s. So we're kind of lucky. So clock is an upstream regulator of two sets of repressors, the period and the cryptochrome genes. So in mammals, there are 3 PERs, PER1, 2, 3. 2 of them are essential, meaning you have to do a double knockout of PER1 and PER2 to get a loss of rhythm phenotype in a mouse. And you can do the same experiment with the cryptochromes. There are only two, cryptochrome 1 and 2. You have to do a double knockout in that case also to get a loss of function. Those two sets of genes are transcribed in the daytime. Proteins accumulate, then they interact with each other physically. They interact with other components such as casein kinase, among others. There are many more. Then this translocates into the nucleus, and this repressor complex inhibits clock B-mal-induced transcription directly by physically interacting with clock and B-mal. Okay. And so this loop takes about 24 hours to complete, depending on the organism, and is what we call the core circadian mechanism. Now, there's much more to the circadian gene network today. There are two other loops, but I've decided not to talk about those today. But they're all controlled by clock B-mal at the top of the hierarchy. And the other two loops, which one involves a nuclear receptor reverb alpha, and the other involves a set of PAR-ZIP transcription factors. Those are all activated by clock B-mal. So to study circadian rhythms, almost 20 years ago, we made a transgenic mouse, a knock-in mouse, where we fused luciferase to the C-terminus of the PER2 gene in order to preserve the regulatory units and to make a fusion protein, because period has a lot of post-transcriptional, post-translational regulation. And so the fusion reporter was a much better reporter than, say, a promoter luciferase fusion. And that mouse, I'm not going to show you those experiments either, but those allow us to measure circadian rhythms at the tissue and cell level using bioluminescence imaging in explant cultures. But one thing that we did is to make fibroblasts from those mice. So this is in this case here, a fibroblast culture that's been immortalized. And then we use bioluminescence imaging. And you can see here, there are about maybe 200 cells that are oscillating. So each of these little twinkling stars is a single fibroblast showing a PER2 luck rhythm. And here on the right are just two cells from this experiment, which lasted 18 days in this case. And you can see at the single cell level, the rhythm is incredible. Every single cell that we measured has this persistent rhythm in this culture. Under these conditions, the cells don't synchronize to one another. They each run according to their own periodicity. There's not strong coupling. In the brain, that situation is different. Again, not enough time to talk about that for the brain oscillator. And so to look at these rhythms here in more detail, if we look at single cell periodicities from an experiment similar to what I just showed you, shown in this an orange histogram, the period at the single cell levels is highly variable. It goes from 22 to almost 30 hours, or 28 hours in this case. But the period is actually very consistent in each cell. It's not like it's a random period, like a cell goes from short to long, back and forth like that. Each cell maintains its own unique periodicity in a very precise way, but the distribution is very broad compared to, say, the mouse, which had this very tight distribution. And so, yeah, so this is just the heat map representation of rhythms for this single cell imaging experiment here, which is 200 plus cells. And they're ordered by initial phase, but you see they all kind of drift over time. They're not synchronized. If they were, you'd see these stripes. Okay, so I had a very talented postdoc, Yan Li, who decided to ask, well, what is the source of this variability? And so what she did is, from this parent culture, she made 150 single cell clonal cell lines. Okay. And then measured the periodicities of those clonal lines, and that's shown in the blue histogram here. And so what you see, first of all, is that there are clones that can be either short or long, and they have a distribution that's almost as broad as the single cells, not quite. And then she took about 20 of these clones and measured their period over 20 passages to see how stable that period was. And in general, the period is stable. It's not like they all went back to 24. So by looking at the clonal variants, and also I'll show you in a minute, by making subclones of a short and a long clone, which are represented here. These are subclones. So this is a short clone. This is orange is a single cell imaging of that short period clonal cell line. And then green, or now blue, is are the subclones made from that clonal cell line. Okay, and so you can see that they're all short on average, but their distribution is also narrower. And then here's a long period clone, single cell imaging in orange, and then the subclones made from that clone. They also are long. And by trying to partition the variance from these various situations, we can then try to estimate what component is heritable, if there is any heritability, which is shown down here. Don't have to go through this equation, but the simple answer is approximately half of the variance is heritable and half is not. It's either random or environmental, okay? And we suggest in modeling experiments that the random component of this is due to transcriptional noise, which we can model in a transcriptional network model of the oscillator, just from the stochastic nature of transcription. That gives you enough noise in the period. So that's one component. To test whether that might be true, we used a drug that Lior what is his name? Oh, sorry. Anyway, that's a transcriptional enhancer of transcriptional noise. And when we did that, the period was lengthened by the drug, but the variance was also increased. So we could increase, if we increase the noise, we could increase the variance. So that's consistent with that idea that that noise is a component of the periodicity. To begin to look at the heritable component, we first exome sequenced these cell lines, and there were no coding mutations. So it wasn't a somatic mutation that caused this. So then we just looked at gene expression with RNA-seq, first comparing just long and short clones, 5 long and 5 short clones, and then the subclones over here. And then you can see the overlap of the gene sets in these various combinations, okay? So there are many genes that were differentially expressed in all of these clones, long versus short, and subclones, long versus short. So there wasn't a very simple answer from gene expression. But we could find many genes that were directly correlated with period, expression level and period. So these are genes that have a positive correlation, meaning as the period got longer, the expression was higher. And then we find genes that have a negative correlation. As their expression gets higher, the period gets shorter, okay? And we validated a subset of those. Those are shown at the bottom. But again, there are literally hundreds of genes like this. And so we think it may be sort of an additive situation. And so if you use WGCNA to look at co-expression networks among these clones, we find a number of modules that are associated with short and long period. So that didn't give us any, specific answer. And so one of the possibilities for how you might get these global changes in gene expression is, of course, DNA methylation. And that might give us a mechanism for how you might have heritable transmission of period from passage to passage in these cells. So we did RRBS to look at methylation, and we could find sites that are correlated with the period, and we could find sites that are overlapping with the gene expression analysis that I just showed you. And so from this kind of analysis, we could then find, looking across our 5 short and 5 long clones, sites that are perfectly correlated with period, okay, as far as their methylation status is concerned. So let's see, where it is. Oh, there it is. So these are 2 examples where you can see a pretty good, or this example here is a perfect correlation with the DNA methylation status for long versus short, many sites like that. But again, we find hundreds of sites in the genome. Perhaps they're underlying these differences in gene expression, okay? And so to test in a more global way, we looked at DNA methylases. And if you knock down DNMT1, you get a shorter period. And if you knock down DNMT3A, you get a longer period. So there's certainly an effect of DNA methylation globally on circadian period. But again, this is acting across many, we think, hundreds of genes. Okay, so I guess the take-home message is the period at the single cell level does have properties of epigenetic inheritance, and that we think that the likely mechanism for that is DNA methylation status, but regulating the expression of many different genes that all contribute a little bit to the period. Okay, so let me get back to the single cell. I'm gonna have to talk fast, I guess. One of the big surprises that came out of the gene cloning was that these genes were ubiquitously expressed across the entire body. Our prediction initially was that it's going to be expressed in the brain, because the master clock is in the brain, and we thought it would just control the body by directing the body, and the body wouldn't have their own clocks. But it turns out that every tissue expresses these genes. And many peripheral tissues actually have much higher expression levels than the brain does. For example, skeletal muscle has the highest level of clock genes. So this has led to the idea that there are clocks essentially in all our major organ systems. And indeed, essentially every cell in our bodies has the potential to have a circadian clock. The majority of our cells actually have circadian rhythms. There are very few that don't. So we think about the body, the circadian system is much more complicated. We're actually a collection of clocks. They're all cellular clocks. And what the brain is doing is it's receiving environmental light information to synchronize the brain hypothalamic oscillator. And then that controls many different behaviors: sleep, wake, feeding behavior, body temperature, hormone secretion. And so many signals are controlled by the brain clock that are available for peripheral cells to utilize as a synchronizing cue. And so we now know that there are many redundant signals that emanate from the brain to the periphery, and each of those is sufficient to synchronize essentially any tissue in the body. The surprise is that the peripheral organs are generally not strongly regulated by the brain. They're more weakly influenced by the brain. I'll show you examples of that in a moment, okay? Okay, so what are all these clocks doing in the periphery? So one example, which was done by my colleague back at Northwestern, he's a diabetologist, and we found that the original clock mutant that we isolated had a metabolic defect. It would gain weight, become obese, have a loss of glucose regulation, and have high cholesterol and high fatty acid with age. And so we studied that. That's a global mutation. But later in this work, what we did was to ask, what is the clock doing in pancreatic islet cells? So we could make a conditional knockout of BMAL in islets, shown here, BMAL in pink, and it's deleted. And when we do this, and we do a glucose tolerance test, we can show a defect in glucose regulation with this conditional knockout only in the islet, compared to controls. And this is due to insufficient insulin production by the pancreas. And so Joe Bass has gone on to show that the insulin secretory pathway is under clock BMAL regulation, and that leads to this defect in insulin production. So that's just one example of a clock function in peripheral tissue. Okay, so if we look at another tissue, and I'm not going to show you all the details of this, but just to show you the summary slide. The liver of the mouse is a major metabolic target of the circadian clock system. And so if you look at gene expression in the liver, you'll find thousands of genes that cycle at the steady state mRNA level. So, about 3,000 out of, say, 12,000 genes expressed in the liver have very strong circadian oscillations. And so we knew that from microarray work and then later RNA-seq. But more recently, in the last decade, we were able to try to identify the target genes of the circadian gene network using ChIP-seq methods for clock, BMAL, and the 4 repressors, PER1, PER2, PRI1, PRI2. And when we did this, what we found is that there's an incredible transcriptional program that occurs every day in the mouse liver, where clock and BMAL and NTAS2, which is the paralog of clock, all occupy DNA in the daytime. They recruit the coactivators P300 and CBP. And along with this, you can see some activation marks in histone peaking here, such as histone 3, lysine 9 acetylation peaking here, K4 monomethylation. And then this is followed by a burst of transcription, and RNA polymerase II occupancy, which you can see here. Then the repressors come in, occupy the same sites. They peak later in the night. So here's PER2, CRY2, PER1 is back there. They have short half-lives. They then turn over, and then the cycle starts over. So what was surprising about this analysis is if we look at how many genes have cycling Pol2 on their promoters, the number is way bigger than 3,000. It's more like 5,000 or 6,000 genes have cycling Pol2 occupancy. And the other surprise is this promoter mark, histone 3 lysine 4 trimethylation, is extremely dynamic in the liver. So it's cycling genes at one time of day, it's not there at all, and then it comes on and peaks, and then it disappears. And so that was a big surprise to us. We can see K4 trimethylation cycling again across 5,000 to 6,000 sites in the mouse liver, huge. So the impact of the clock on not just transcription, but on chromatin state is huge and much bigger than we had thought before based on just looking at RNA expression. Okay. So what are these target genes? So just one example, the top go category is metabolism. Here's the KEGG metabolic chart of all metabolic pathways. And overlaid in red are all the clock female target genes. And so what you can see is the entire chart is covered. Every fundamental metabolic pathway in the cell has direct transcriptional targets regulated by the clock system. And this picture is not unique to the liver. We can see this in the brain, skeletal muscle, pancreatic islets, every tissue that's been looked at carefully with ChIP-seq gives a similar picture. And so I think this is telling us that the clock is doing something very fundamental in a cell autonomous manner. It's fine-tuning metabolism every day in many tissues, not just the liver. Okay, so let me go to the final part of our story, and that has to do with what is the role of clocks and timing as far as aging and longevity is concerned. So I've already told you the clock regulates metabolism, but metabolic pathways feedback and also regulate the clock directly. So this is just a couple examples. The clock regulates NAD levels. NAD can regulate the deacetylated SIRT1. SIRT1 can deacetylate BMAL and HER2 directly into the core mechanism. Another example is AMP kinase, a nutrient-sensitive kinase, which phosphorylates cryptochrome proteins that regulates their stability and their turnover by the proteasome. And so the clock not only regulates metabolism, but it's embedded in metabolic pathways and is influenced by metabolic factors directly. So why did we get interested in time of feeding? So here's a very simple experiment done by my colleague Fred Turek a long time ago. We all know that if you give mice a high-fat diet, they become obese, like 6 mice. Okay? So what they did was just to restrict the high-fat diet to either 12 hours in the daytime or 12 hours at night. And what happened is the mice that ate in the daytime, shown here, which is the time that mice usually sleep, that's the rest period, they became fat. But the mice that ate during the night, which is their active period, did not gain weight, or as much weight, I should say. They both ate the same amount of diet, and they had the same activity level overall. So the mice are metabolizing this diet in a completely different way, depending on what time they are actually eating the diet, okay? This kind of experiment has been replicated by many labs now. This is the initial first replication. This is ad lib, high-fat diet, mice get fat. In this case, if they're restricted to an 8-hour window at night, they don't get fat, shown in red, okay, here. And many other versions of this experiment have been done. So these are just a couple examples of initial replications of this using different kinds of diets: high fat, high carbohydrate, 5 on, 2 days off, all sorts of combinations. As long as they were restricted to eating at night, they're protected from weight gain. And in addition, also there are health benefits, better glucose regulation, and other measures. And there's now actually quite a bit of work in human trials looking at time-restricted eating to see whether there are benefits. And in general, there are clear health benefits on short-term trials, usually measuring body weight for weight loss, but also measuring glucose regulation, insulin sensitivity, things like that. And so far, overall, in cases of overnutrition, these are all experiments where the diet has very high caloric content, okay, and then you restrict the window, and that offers you protection. So why did we get interested in aging? Well, when we looked at the classic aging experiments, these are shown here. What's been known for a very long time, at least 80 years, almost a century, is that if you reduce calories in rodents, they live longer. Okay, and in this work here, it appeared it was proportional to the amount of caloric restriction. The more you restrict the calories up to a certain extent, the longer the mice would live. Okay, but if you look at how they fed the mice, this is their standard protocol. They actually have a methods chapter that describes this protocol. They fed the mice 3 times a week, Monday, Wednesday, and Friday. Okay? And they fed them in the morning. So for circadian biologists, this is not a normal feeding pattern for a mouse. Mice eat every day. And they also eat most of their food at night, as I'll show you, not in the morning. And if you feed at the wrong time, this has a big effect on peripheral organs, such as the liver. So we were wondering what effects these, to us, funny feeding protocols would have on aging and longevity. And so we were able to get a grant to do a longevity study to try to understand what is happening if you manipulate the feeding pattern. But to do this experiment, we had to develop an automated feeding system, because the reason in the original experiments they only fed them on Monday, Wednesday, and Friday is the experiment takes 4 years to complete. And most of your staff don't want to work 7 days a week. Much less put the food on and take it off twice a day, 7 days a week, which is what you have to do for time restriction. So it became clear that nobody in my lab is going to do that experiment either. So we spent a couple of years, we developed this automated feeding system. The reason we had to do it ourselves is the commercial solutions are either too expensive or too big and don't give us enough control. So we can't have enough units to do a longevity experiment, okay? So this is done by Vicky, whose picture was just there. And here is an actogram plot showing activity. The mice are on this light cycle, they run at night, and then the pink dots are when they take a 300 milligram food pellet. And so this is an ad lib fed mouse, and they eat 3/4 of their food at night, but 25% of their food in the daytime during their rest period. When we put the mice on caloric restriction, In this group, they had access to the daily allotment of food beginning at time 0 dawn, okay? As they got hungry after about a week or two, they ate all the food as soon as it was available in less than 2 hours, okay? And what this means is that they then went into a severe time-restricted feeding protocol that was voluntary, where they eat in 2 hours and they fast for 22. Okay, this is also true if you begin feeding at the beginning of the night here. Okay, so when you reduce calories, you're not just reducing calories, you're changing the feeding behavior of the mouse. And the mouse adopts an extreme time-restricted feeding pattern and has very long fasting intervals. And we know all three of those factors are good and potentially beneficial to lifespan. So we started a survival study. We have the 3 groups I just showed you, okay? And then to get around the binge eating, we stretched the food out over 12 hours. We doled it out so they couldn't eat it all in 2, day or night. And then importantly, we had a 5th group where food was given continuously. So there's no day-night difference and there's no fasting interval, okay? These are the activity records of single mice in each group over 3 years. Overall, they all remain nocturnal, even if they're day fed. But you can see that in some instances, day feeding causes more activity in the daytime during the rest. Okay? And then this is the feeding pattern, which remains consistent over 3 years in all these groups. And the mice are very obedient. They eat the pellet as soon as we give it to them because they're hungry. Okay, so then this is the food intake of the ad libs in gray. They actually increased their food intake once they became older. And initially, the calorically restricted mice were restricted 30% from the ad lib at the beginning of the experiment, and then they were clamped at this level for the rest of their lifespan. This is the activity level of the mice on the running wheel. It gradually declines as they get older. And then this is the body weight of the mice. The ad lib mice gradually increase weight until they're about 20 months of age, spontaneously lose it. Okay, so they get pretty fat. They're approaching 50 grams, whereas the calorically restricted mice all maintain low body weight, but they don't lose weight, importantly. Okay. So here's the important part, survival. So in gray is shown the ad lib group. They live just under 800 days median lifespan. That's normal for black 6 mice. And one of the big surprises is this group in purple. That's the continuously fed group, but they're calorically restricted. They only live 10% longer. When I asked the aging field, my aging colleagues, what do you predict is the most important factor? They all said calories. Doesn't matter what you do, you reduce calories, they live longer. So this is a big surprise because we've reduced calories, but they only live 10% longer. Okay. If we look at the day-fed mice, they have a fasting interval now. They live another 10% longer, 20% total. So fasting does introduce another benefit, another 10%. But the mice that eat at the right time, that is at night for a mouse during their active period, they live 35% longer median lifespan. So the only difference between the day and the night groups, is the phase at which they eat. Everything is identical. So alignment with the circadian clock of the mouse is extremely important for lifespan and gives you this 3.5-fold benefit over the spread group here. Okay, so we're super excited about this result. because it suggests that the clock system is really important for longevity as far as alignment of your feeding time. So what did the mice die of? They all essentially they all die from cancer. And the primary causes are lymphoma and hepatocellular carcinoma. Most of the lesions at necropsy occur in the liver, as shown here. And the mice that live longer don't die of a different spectrum of cancers. They just have a delayed onset of all of these cancers, okay? So we began by looking at the liver because that's where the most lesions were. And we first compared younger mice with older mice, 6 months old and 19 months old. Okay, so we use 6 months because they're fully adult. We use 19 months because they're old, but they're not decrepit, meaning 90% of the ad lib mice are still alive at 19 months. At 24 months, that's not true. A significant fraction died off already, okay, if you wait longer. And so just looking at the ad lib group, with age, 2,000 genes go up and about 500 genes go down in gene expression, okay? So the genes that go up are all involved in inflammation and immune function. There's only one set of go categories. It's incredible. And you can see it's highly significant, okay? The genes that go down are all in metabolic pathways. And again, it's a very uniform gene ontology picture. It's quite interesting. Okay, so then if we break this down to just percentage deviation, so ad libs had a 14% increase, a 4% decrease, and we look at all the other calorically restricted groups, what we see is that there's a general correlation with lifespan. The longest lived groups, these two here, have the smallest differential gene expression, meaning they have fewer inflammatory genes that go up and fewer metabolic genes that go down, okay? If we ask what genes are protected just by calories, it's the large majority of them, 1,200 genes. We introduce a fasting interval, we see another 150 genes or so. And then the longest lived group reduces this to a subset of about 68 genes. And the perfect example gene here is interleukin-1 beta, where there's essentially a monotonic decrease in correlation with lifespan as an indicator of inflammation status, I think, in the mouse. If we look at circadian gene expression with age, what we find is there's a decline in the amplitude of cycling genes. This is known in the field already. If we look at fold change across the whole genome, there's a significant decrease in fold amplitude. So with age, cycling genes are declining in amplitude, and that suggests that the circadian drive of the clock system is decreasing as the mice get older. The other change that we see is there's some genes that don't change in level, but their pattern is completely altered by the feeding status, so especially daytime feeding, which disrupts many genes, some of which are classic aging and longevity genes, such as IGF-1, shown here, FOXO3, and AMPT. Okay? So to sort of summarize, we find that time of feeding is really an important factor in determining how effective caloric restriction is in extending lifespan. And it's surprising that it makes such a huge difference. And overall, we see activity, metabolism, inflammation, those are decreasing, inflammation is increasing, cycling genes are decreasing, and all of these are partially rescued by caloric restriction. The other interesting feature is if we look at classic aging pathways, such as IGF-1 signaling, mTOR signaling, AMP kinase, these are all under circadian regulation and are direct targets in many tissues. So here, this is just a summary shown in purple by tissue, whether these particular genes are actually under circadian control. And so you can see many of the classic pro and anti-aging gene pathways are under circadian regulation. And so we're working on the hypothesis that one way to intervene and try to rescue gene expression that's declining with aging is to try to rescue circadian transcriptional drive, because it's upstream of many of these pathways. And so by attacking one nodal point, we may influence many different aging pathways simultaneously. And so we're doing a number of different interventions currently. Time-restricted feeding, which rescues circadian amplitude. We're using a genetic intervention by expressing clock genes or clock the gene in transgenic mice, which boosts expression. And we're looking for small molecules that enhance circadian function. So I'll stop there. Thank you.
Thank you, Joe, for a fantastic talk. Now, the only thing that's streaming in terms of the microphone is from here. So, but I don't think there's any point me running around with a microphone. So if you just say your question loudly, I'll then repeat it and then Joe can answer. So Anna. Oh, you can stay there and I'll repeat. Oh, you're going to say it.
Really fascinating talk. Thank you very much. Really enjoyed the talk. Got a couple of questions. One relates to the circadian rhythm in the cells. So in culture, of course, they do their own thing. In the body, you said it wasn't really regulated much by what happened in the brain. Mm-hmm. But can I assume that it's still somehow synchronized, maybe by other things, by activity, so that during the activity period at night for mice, during the day for humans, mm-hmm. All these metabolic enzymes are more active to support the activity?
Yes, yeah. So, I didn't mean to say that normally the animals desynchronized. Normally we're synchronized because everything being controlled by the brain, such as feeding, is introducing nutrient cycles, which are then synchronizing, say, the liver. And so normally everything is in sync, as long as behavior is under appropriate rhythmic control, meaning eating at the right time, for example.
And one other, so with the longevity study, so one group that you didn't have was calorie restricted, not calorie restricted, ad-lib, but time restricted, and I think you're doing that now. But the activity levels of the calorie restricted mice, did they go down?
Yeah, they go down with age, but they do have higher levels than the ad-lib group.
Really?
Yes, but the confound there is the ad-lib mice are fatter. So we can't eliminate the possibility that their activity is lower because they have higher body weight.
Johannes.
Great talk. I was wondering, I don't think you have done the short experiment. What if you change the periodicity of feeding so you don't feed always at the same time, but it's random when the mice different? So, but I'm thinking whether the mice get used to being fed every 12 hours. Maybe you see that in the data that they stop running before that next 2-hour feed in order to prepare. Maybe the metabolism in the mice is also preparing to getting used to that 2-hour feeding periodicity. And I was wondering what you expect the experiment to show you random feeding.
So the question is, do the mice get used to when they're going to get and kind of begin to expect when they're going to get the food, or can you have random feeding times?
Yeah, so that's a great question. So I have this new postdoc who's coming from an aging lab, and that's her burning question. And so she's actually going to do that exact same experiment. Have mice under caloric restriction, but feed them at random times to see whether this habitual pattern, as you ask, is important. My own personal opinion is that, you know, it's hard to completely randomize feeding time, because then that means you're going to feed at the wrong time. And I think that that'll be detrimental. So we'll see.
Melissa.
Hi.
Hi.
When's the point of no return? So if you have an ad lib bit, now it's half of its life. Can it turn things around?
Great question. So can you turn it around? So can you start intervening in the middle of your life, for example?
Yeah, so yeah, we haven't done this experiment, but other people in the aging field have initiated caloric restriction at different ages. You know, say 6 months of age, 12 months, 18, and 24. And it's kind of interesting. So you can see a benefit at 6 and 12 months, okay, middle age. But at 18 months of age, there's almost no benefit. And at 24, it's detrimental. The mice die sooner. So.
Was Andrew, then here, then Wai Hong, and then that we should probably finish questions at that point. Yep. So Andrew.
You mentioned that DNA methylation changes with circadian rhythm, or at least with circadian rhythm, and DNA methylation is a predictor and potential contributor to aging. I was just wondering if you were looking at DNA methylation as maybe mechanism.
So the question is, DNA methylation changes with age and might have some determining factor, and what's the relationship, I guess.
Yeah, so we've looked at DNA methylation in these mice in this experiment with Steve Horvath, who's used to be at UCLA, now at Altos Labs. And of course, you know, Steve is well known for the DNA methylation clock to determine biological age. And in this experiment, we can see a clear difference in DNA methylation between ad-lib fed mice and the 5 calorically restricted groups. But his clock so far hasn't differentiated the lifespan differences within the calorically restricted groups, you know, that I showed, going from say 10% to 35%. So we're trying to work with Steve to look at the original DNA methylation data and see if there are sites that give us a much better measure of biological age in this experiment. So that'll be a special clock for these mice, you know. But it could be interesting, because maybe it'll tell us something about loci in the genome that would be interesting to look at.
What's the effect of the mouse house being open, or like people working in the mouse house during the day, and rather than, you know, working at night?
You mean in a regular facility, or? Yeah, because I mean, our mice are in a separate facility, and they're in enclosures, so they're not disturbed by the normal animal staff working in the daytime. But certainly in the normal facility where all the, you know, cage changing and everything else going on in the daytime, it's very loud and noisy. It definitely disrupts the mice. I think one consequence is if you have certain strains that are hard to breed, it could actually make it difficult to breed those mice. But that would be the most direct consequence that we would typically see.
Wai Hong.
So we could have circadian rhythms. How do you think of all the cells that are in that microphase, and what is the, you know, why are they not regulated?
So I forgot to say the last question, but this question is, do microbes have circadian rhythm, and could their rhythm, if they have it when they're in your microbiome, wouldn't they be giving off metabolites, and that might influence our rhythm, if I get it right?
Yeah, so microbes definitely have circadian rhythms. The best example are cyanobacteria, free-living bacteria have extremely good circadian rhythms. And there's more and more evidence that other types and strains of bacteria can have circadian rhythms. In the past, it's been difficult to show that they, have circadian rhythms. Now, in the gut, the microbiome clearly has diurnal variations. And the initial work done on that suggested that that's largely driven by the feeding cycle. But there are other groups that have provided evidence that some of the microbiome species actually have their own clocks also. So we've done 16S sequencing of this experiment too, but we're still analyzing the data. But it's pretty interesting. You can definitely see just by eye, it's all cycling. And that the feeding time has a pretty strong influence on that.
Well, we've grilled Joe with lots of questions. We're about to grill him with some more, but let's thank him for his seminar. So the idea now is that we could obviously chat to Joe all day about circadian rhythm, and that's great. But what we wanted to do is to have the next part to be about genetic screens. And so coming up after we have some Q&A time, we're going to have the 5 students that are going to be pitching for some funding to do a genetic screen. But to begin with, what I wanted to do is take advantage of the wealth of experience that we have in Joe and also Marco Harold that you'll all know from WeHi, and the huge experience they've had in designing and running and then following up in vivo and in vitro genetic screens. So I'm going to start with some questions, but then I would really love it if everybody else also asked some questions. And so we can, I would imagine any sorts of questions, because they've had to come across lots of problems as they've run lots of genetic screens in their lives. And so what I will do is, unless it's specifically directed to one of them, maybe you can each give us your opinion. So I'm going to start with one, which is, what do you think how do you weigh up spending longer and longer designing the perfect assay for your screen and just getting started so you feel like you want to start the screen? Do you need it to be perfect before you start?
Who wants to go first? Okay. So, I would say that it's really important to understand your phenotype. So that's going towards this perfectionist side, but you can overdo it, I guess. Huh? So I don't mean that you have to be completely perfect, but it's essential to understand what the important phenotypes are in your screen. So to give you a circadian example, okay, we can, in that wheel cage, we can measure lots of things. The activity level, the phasing, the day-night difference, whether the activity record is fractionated or solid, there are all sorts of quantitative features we can extract from those data. But for the circadian clock, those are all what we would call output pathways that may or may not be controlled by the circadian clock. In fact, we know some of them, like activity levels not really directly controlled by the circadian clock. So it turns out that period is the most important phenotype if you're trying to find a clock gene, because it's that phenotype or output is not masked by all the factors, between the clock and what you're measuring, the feet of the mice, and it's very hard to alter the period without affecting the clock. You see it, because it's a timing thing or rate thing. So I think that's a really important thing to think about in your screen. Is there some key parameter analogous to period in an oscillator that you can try to focus on in your screen.
That's a great answer.
Yeah.
Marco, how about you?
Yeah, I mean, I'm a bit similar. I think that a screen, the readout has to be clear from the beginning. So if it is clear cut what you can get out, then these screens don't need to be very complicated and you don't need to establish too much. The simpler, the better. The simpler, the more you get out of a screen. The simpler, the less complex your tools need to be. And the simpler, the less powerful your tools need to be. So I think, and if you have a simple screen, you get learnings, which you can then apply to more complex scenarios and get more and more complex over time. When we think about our history, we started with very easy screens, dead live, knockout screen. And now we're going to the screening where we have single cell readouts with CRISPR activation in vivo. So I think, but this wouldn't be possible if you have never done the first screen. So I think about the easiest readout and do your screen without thinking too much about which tools to apply or how complex it could be.
I'm going to ask one more and then hopefully we'll have other people thinking of their follow-on questions. So when is the time that you say, I've found enough things from a genetic screen and you don't do another genetic screen? And in fact, you just focus on the thing that you found rather than dreaming up another cool genetic screen as a subsequent screen to find modifiers of clock, for example, or modifiers of your modifier of clock, which I would do because I love genetic screens. But how do you, again, and I was asked this in a fellowship interview, when will you stop screening? And I thought, I don't know the answer to that question. So I'm asking both of you.
I think that comes back to what I just said, the complexity, because the more complex we can be, the more refined phenotypes we can find. And so I think you will not really stop screening, because if you see the probably simplistic screens and you look for different phenotypes, you will find these few phenotypes. And then you can follow on a few of your genes. And what is very common in the whole screening community, nobody or almost no one will touch something very unknown. Everybody will know what they know already a little bit and follow these targets. But I think then you don't stop, you just keep on going because you want to more understand how these few targets we understand and know are, for example, regulated, how they play a role in different scenarios. And so the answer is never.
Joe, how about you?
Yeah, well, I think with mouse screens, which are, pretty expensive.
And long-term.
Yeah, and require space, you do have to be practical and, and decide whether the mutants you've isolated are sufficiently interesting that you're going to invest in them first and not screen. And so, and again, that has to do with knowing your phenotype. How extreme is your phenotype? So with clock, we knew it was extreme because it had a 28-hour period. That's as good as the fly mutant, right? The most extreme fly mutant. And so we focused on clock. So that's just, a real example. But I think it's in mouse screens, you really do have to trade off cost,
yeah.
Unfortunately. Yeah, and time, definitely time.
Okay, I'm going to throw it open now. Johannes.
It's my question to the group, but what is your opinion of organ screens? Having, it might not happen a lot, but having two screens that work together from different directions, it might seem quite powerful. I'm sure that's
I'm rather than repeat it straight up, I'm going to say, can you give an example of what you mean first, and then I'll repeat it. I'm not sure if I understand what you mean.
It's kind of like having two screens that work differently, but trying to tease out the same things.
The screen and cells match for them. Screen and fine.
Yeah. Okay. So the question for those people that are streaming is, what are your opinion on having two orthogonal screens, but screening essentially the same process, but in different ways? Is that a fair summary? Yeah.
Well, I think if you have, it comes back to money. If you do it in mice, of course you could do. If you have enough, you could do it. The more you can kind of validate in the beginning what you want to follow up, the better. But then it also gets cost-wise quite expensive and a lot of labor. So probably best to start with one screen and then extend maybe a reduced level of screening onto multiple aspects, meaning on multiple organisms or multiple scenarios. So that would be my recommendation. Instead of using a CRISPR-A and CRISPR-I screen in vivo or something like that, do one and then look at the top targets and validate them with other methodologies.
Yeah, so I can give you an example that we attempted. So instead of using running wheels, we decided to look at the HER2 luck mice. where you can just clip the tail tip and measure the rhythm of the tail tip. Okay, has a beautiful rhythm. And so the person who made the mice wanted to do that screen. So I go, fine, you try it. And what she did was she measured the bioluminescence rhythm from the tail tip, and then also put the mice on running wheels. So we had both phenotypes, and she tested 100 mice that way. And it was very clear that the running wheel assay was way better than the bioluminescence assay, because in the bioluminescence assay, you can't measure the period very accurately, only to within maybe half an hour or something. But on the running wheel, we have much better determination of the circadian period. The variance, the standard deviation in the onset of activity or for the period in a mouse is less than 1% of the mean. It's better than any biochemical assay,
Astounding. So we did that. So we did an ENU mutagenesis, or I did an ENU mutagenesis screen. And then 15 years later, Andrew and Tash and I did the same sort of, the genes that came out, only the genes that came out of that screen rather than the ENUs, obviously genome-wide. We only tested them in an exon activation screen, both for silencing. But we went the other way around, right? So we did whole genome and then went, well, actually, this is in theory a simpler assay because it's in vitro, but we only then screened a subset, which was then very powerful, but it was definitely one followed by the other. Melissa.
So in cancer screening, the drug screen, the small molecule screening, there's a Z-score that you calculate for any of your assays. Do you think a similar score exists for genetic screens, and do you employ it?
Yeah, I mean, you can. We tend to, we do controls and look at the distribution of the phenotypes, and then we typically have a three standard deviation cutoff, Z-score of 3, which is similar to the Z-score in a chemical screen. Yeah.
So called the Z-score.
Yeah.
Yeah.
We don't. So we just, no, we are using the analysis. It's a different analysis pipeline. We're looking for enrichment depletion. And that's essentially it. So we're not.
Because yours is a genomic output rather than I have an assay that is measuring the amount of GFP or whatever, or amount of, the actual period. Yep.
Eva, when you do a screen, you say for sort of I guess having control to how we use what is the term have and control
The question is, what do you do if you don't have a control for your screen because you don't yet have a positive regulator or negative regulator of that person?
Yeah, I mean, as I said, we just look at the wild type distribution of your phenotype as the main parameter you have to pay attention to is how good is that Z-score in your screen.
Find an outlier, basically.
Yeah, so we didn't have a positive control, ahead of time. That's correct.
Yeah,
right.
I think that's a bit different as a chemical screens where you have something which completely obliterates your cells and something which keeps them alive, for example. So in some genetic screens, we don't have any clear positive control and you have to do it. And within like in our case, CRISPR libraries, I mean, you have often the ones we are using nowadays, the 10, 15 guides for CRISPR-A. And then we know that if you have 5 or 6 enriched, we can be relatively assured that it is a real hit. So that's more our kind of, and then we pick out at some point, something reoccurs and occurs and occurs. That will be a positive control in the future.
Andrew.
I was wondering, after your screen and you've got your hits, I would prioritize them. Based on the data.
Yeah. So the question is, how do you prioritize your hits?
Depends how many PhD students you have. No, I mean, It's very, I guess, if it's sometimes it's so clear cut, the first 5 hits and then there's long nothing coming below them, then you know how far you go. And sometimes we set it's an arbitrary cutoff, essentially how good the screen looks. We had recently a screen of 2,500 transcription factors, which we narrowed down to 200. It could have been a bit more stringent, but we said, okay, we make a smallish library and test the next 200 and then go down to the next 30 and so on. So I think that's the way we pursue normally any of the screen.
But would you favor a completely something that's classified as a transcription factor, but that nobody knows anything about? Or would you favor one where somebody already knew something about it? Maybe there were some antibodies available and a mouse, and
Yeah, so that's what I discuss a lot with colleagues who do that, and they said essentially, and that I alluded a little bit in the beginning, you follow usually where there's something known already in the beginning. And if you have enough manpower, you could follow these unknowns, but it's very dangerous because they could be really false positives and you would spend 5 years of your time in it and then it's like, oh, there was nothing and that's it. So I reckon it depends a little bit on that.
Yeah, so I think the cutoff depends on the type of screen again, obviously. That's sort of self-evident, I think. But as far as pursuing hits, I agree. It's much easier if you can connect that gene to some other pathway that you know about already. But, in some screens, you may know nothing. And so you have to go and pursue something that is completely unknown. And in a way, if you're lucky, that could be extremely exciting because you might uncover something completely new.
So you get a list of hits and that's part of it, but re-screen in the time of the library against the hits and then re-screen the same hits against the
the question is about rescreening. So I think Marco just said that's what you're doing.
That's what we're doing, exactly that.
Yeah, we do that for, say, CRISPR screens, but not for a mouse ENU screen. Yeah, I mean, the way we validate mouse ENU screens is we try to find the null mutant because there's a knockout mouse project where they knocked out every gene in ES cells. So in the old days, you could then try to get that mouse. Today, you would just make the knockout. In fact, what we do now today is we make the mutation with CRISPR and recreate the mutation. And that's the most efficient way now for mouse ENU.
Are there any other questions? Or otherwise, we'll move on to student questions.
I have a similar question to the previous. Anyway, when we do the design the genetic screening, do you think how many times we need to repeat the experiment, then we go to confidently go towards our data? Because theoretically, you would the ones you go to some positive hits, oh, that's perfect, and then you repeat it again, and the hits disappear.
You're talking. So the question is, how many times should you replicate your screen? So I think you're meaning an in vitro screen, probably rather than an in vivo ENU screen, or even in vivo CRISPR screen. Anyway, not an ENU is different in that case, but yeah, screen replication.
I think it depends on the quality. So we see that if you have a very good starting point of representation of guides, Cas9 activity, there's no need to repeat it because you can be very reassured what you find. You will validate it anyway. You're not letting these things go and say, these were our top hits that are that's what I believe and that's what I publish, or that's what I will work on. We first do a validation round, and I think you don't need to necessarily repeat genome-wide screen a lot, 2 or 3 times.
What's up?
There's a lot of databases and a lot of new techniques coming through. You find an unknown protein, and you've got a pretty good structure of it, like alpha-fold. How do you see all of these new bioinformatics and databases contributing to screening? Are we in a world where they're much more powerful now?
I think it needs some people who can integrate all this with each other, and then it's very, very powerful. But I think that we miss system. I think there's people who have expertise here, there, and there, but these connections are not there yet. So I think we're looking into another 4 or 5 years until this is really happening.
Laura?
I don't know, maybe
really general question, but what is the thing that you like to improve in space, like the main issues of the technique that makes you feel like, oh, I have to screen, but it's a really issue.
Yeah, the question is, what's the bottleneck, or what,
Yeah.
what needs
No,
to
I
be
have,
improved?
I have an answer for ENU, but
So for ENU,
yeah, for ENU, it's you have to validate that mutant somehow. So and you want to know what the candidate gene might be. And so my colleague Bruce Beutler at UT Southwestern is doing an extremely clever screen. So with ENU, you make we call a G1 mouse. That's the offspring of a mutagenized male mouse. And you use that G1 mouse to found this 3-generation pedigree, okay, to make the mutation recessive down here. 1/8 of the mice will be homozygous mutant if they're viable. The problem with that, so that's what we did 20 years ago. The problem is then you have to take that mouse and try to map the mutation, okay, which is extremely inefficient because you have to make another set of crosses, okay, and then do linkage analysis. So that killed us back then. So instead, what Bruce does is he takes the G1 mouse, he exome sequences it, and he determines all the point mutations in that mouse. And if it has enough, it carries, his threshold is 50. It has to have at least 50 coding mutations. Then he breeds the mouse, makes the pedigree, and then he genotypes the pedigree with those ENU point mutations. And so he can immediately see linkage in the pedigree.
he's done his linkage cross already.
Right.
Basically.
No, it's brilliant. So that was a solution to, I would say, the biggest bottleneck that we have in ENU is the mapping.
CRISPR screens, it's a quality. I think we see a quality. I think there's a lot published which is not high quality. And the analysis, I think everybody uses magic, and unfortunately there's not much more out there, but it's not the best. No.
Harry.
Question online from Shubi. He says, I've never done genetic screens, but my understanding is there is a very degree of saturation within the gene, especially for loss of function screens. Are there any new genetic screens available that use new technologies to achieve 100% saturation?
It's great.
So that was Shabi's question. So that, and I mean, I guess people can read that online if they're online, but basically, can you achieve saturation genetic screen or new ways?
Well, I think you can with CRISPR, you know, in cell-based screening, no question. In a mouse, it's a little harder.
Yeah, but if you keep hitting the same gene repeatedly, then
Yeah.
you end up thinking it's probably you've got to close to that. You kind of estimate you're at saturation.
Right. And then in this Beutler screen, we actually can look at the hit rate just at the nucleotide level across all our favorite genes. So he's not at 100% saturation, but he was already at like 60% or something. It's pretty high.
This is going to be the last question. I'm actually going to go down here to Ollie instead. Sorry. And then we'll move on to the next stage. So last question, Ollie.
Okay, so I'm not really that experienced because I just complete my stats by genotype in vitro screen and kind of closing up like what is best to prioritize. So when you complete the screen, obviously select a couple candidates. Assay could also screen in a different cell line, which would be beneficial, or you could use some targeted library. So I'm just wondering how you come to that decision of like what to prioritize next and how to essentially proceed with the experiment.
So the question is, you've got a few hits. How do you proceed? Because there are too many possibilities of what you could do next, basically.
So in vitro, what I would do, I would use exactly these guide RNAs and test first in the same cell line under the same conditions, whether they reproducibly give you the same phenotype. And then if they do, then you can extend, if you have certain genes, to use more guides if you haven't had already, and then test those ones, across multiple cell lines and use the same assay. And then you can be more elegant and more complex into other assays. That would be my take.
Great.
Yeah.
Well, so I think that probably so that we are going to try and get back closer to time, we're going to stop there for the Q&A part, and we're going to switch over and switch gears to the pictures. So Marco and Joe can come and sit back over here, and then we'll invite up Matthew for the first of the pictures. So if you want to come up, and you should just be able to press next slide.
Hello, everyone. My name is Matthew from the Night Lab, and today I'll be talking about a novel class of time-resolved genetic screens. Let's start by looking at a recipe for a grilled cheese sandwich. Like any good recipe, if you follow these steps in the right order with the right amounts of ingredients, you get a perfect sandwich. What happens if you change the order of the steps and ignore the amounts of ingredients? If you follow this recipe, you get a different kind of sandwich. Now imagine a future where we use genetic recipes to change a person's cells to any cell type that they might need. Here are two examples of recipes where the order and amount of gene expression is important. Firstly, in a class of early hematopoietic stem cells called common lymphoid progenitors, if you transduce these two transcription factors, you get either basophils or eosinophils, depending on the order of transduction. Secondly, in cardiac fibroblasts, if you use these transcription factors, you get varying efficiencies of reprogramming to cardiomyocytes, depending on their relative levels of expression. By screening the order and stoichiometry of transcription factors added to cells over time, we may be able to determine the genetic recipes for all possible cell types. However, current methods to screen different genetic perturbations to cells can only track which perturbations are expressed by each cell, but not the order in which they're expressed. So what if you had a way to directly record the perturbations experienced by cells over time? We and others have developed a method to do this using prime editing, a form of CRISPR that allows you to write specific sequences from guide RNAs into a target site in the genome. By attaching guides with barcode sequences to different perturbations before adding them to cells, we can track the expression of perturbations in each cell over time. All of these guides will target the same site in the genome, which you can choose beforehand to be the recording site. Each time a perturbation is expressed, the corresponding guide adds a barcode to the end of the recording site at a frequency proportional to the stoichiometry of that perturbation. This means that barcodes will accumulate at the recording site in the order that the perturbations are expressed in each cell. So, for example, to screen differentiation programs, we can deliver guides with barcoded sequences on the ends of transcription factors in a pool of mRNA together with the prime editor. We can dose cells with this pool of factors at multiple time points using lipid nanoparticles, so that each cell receives random factors at different points in time. The transcription factors expressed by each cell will be captured as barcodes in the recording site. For any given cell type at the end of the screen, we can sequence the recording site to determine the program of transcription factors required to produce that cell type. This approach has three advantages over current genetic screens. Firstly, we achieve massively higher throughput by permuting genetic perturbations. With only 10 perturbations across 5 time points, we can explore 30 million more genetic recipes than the same number of perturbations expressed combinatorically. Secondly, by recording perturbations into the genome, we get a continuous log of perturbations experienced by all cells and their ancestors across generations. Finally, we can read the perturbations from the recording site, either by bulk DNA sequencing after sorting for one target phenotype, or we can explore the programs for all possible phenotypes through single-cell RNA sequencing if we allow the recording site to be transcribed. As an estimation of budget, we can replicate the 2006 Nobel Prize experiments involving the induction of fibroblasts to pluripotent stem cells by screening different permutations of the Yamanaka transcription factors. With double-stranded DNA from IDT and plasmids from AGI, we can in vitro transcribe the original panel of factors with barcoded guides as mRNA and deliver them via LNPs. Using this protocol here for reprogramming human fibroblasts via serial transfections with mRNA, we can generate stem cells after 2 weeks of culture and then sort them by fax. Finally, we can sequence the recording site to determine the transcription factor programs that most efficiently produce the stem cells. Excluding routine material and labor costs, the total budget will be under $10,000. Thank you.
That was a very lovely talk. So I think we can do 1 or 2 questions each. So are there any burning questions? Johannes?
How do you control for what you put into the cells? Like you add mixture of guides or unedited guides at different times. How do you control for what you add and what's kind of representative that you add?
Yes, so I guess the question is, how do we control the different perturbations that are being added to cells in this screen? So essentially, the way that we can design it is we can have single constructs that pair a perturbation to a uniquely barcoded recorder guide. And then what you can do with this is you can generate a pool of different perturbations and then package them into your vehicle of choice, in this case, LNPs, at an MOI of 1 or less, for example. And then this means that you can control the number of perturbations that get into each cell at each induction point. And all you would be doing is inducing the cells multiple times with this pool of factors, so that cells will get 1 perturbation per induction time. Yeah.
Daniel?
I love that talk, Andy. Excellent job.
Thank you.
The examples that you gave, they tended to be master transcription factors that we know regulate metabolism or IPS. So how many of those are there? That's probably the substrate of your screen. And the second question, how many unknowns might there be to discover with an approach like this?
Yeah, so the question is about, firstly, how many known transcription factors or regulators of interest there are, and secondly, how many unknown equivalents would there be? And so I think firstly, with this kind of screen, a proof of concept experiment would kind of look at the unanswered question of what the optimal ordering of known factors is. And for example, with the Yamanaka factors, with just 4 different candidates, people have shown that if you express these in cells at different stoichiometries, for example, OSKM, if you express OCT4 at 3 times higher levels than all the other factors, you can get up to 1.5 times greater efficiencies of reprogramming to the end state, which in this case is the iPSCs. And then if you change the ordering, you also get similar effects. So with this screen, you can basically answer that question. But then in terms of like unknown regulators, I guess you can do the same thing as well. The good thing about this is you explore both the combination of factors that you choose to explore and the ordering of those factors over time. Yeah.
Last one, Ollie. Yep.
So obviously, I'm not too familiar with transcription factors, yeah, et cetera. This is more of a holistic question. But my understanding is that like during differentiation, you have transcription factors that are involved earlier on, and they come back later in the screen. So obviously having this binary process without the potential limitation, because depleting one of the transcription factors earlier on the screen, you consequently depleting a whole bunch of others. But you're not really accounting for the fact that they may need to return as part of that process.
Yeah, so the question is about the 2 different ways that transcription factors can kind of interact with each other over time. One is they either turn on in a binary fashion and turn off, or 2 is they additively stack over time. Okay, so I think what the advantage of this approach here is you can choose whether to transiently or stably express your different perturbations, so to speak. So in the case where you're interested in something that is likely to be an on-off system, then simply transiently inducing the perturbations in the cells would work. But you can also definitely stably express each perturbation, perhaps with different pools of perturbations across time so that they stack across time as well. Yeah.
Okay, thank you very much, Matthew. And next we'll move on to Yin Yuan.
Yeah, thanks. Hi everyone, my name's Yin and I'm a PhD candidate in the Robertson Way Lab in the BC division. Thank you for the opportunity to pitch our idea for a CRISPR knockout screen looking for potential tumour suppressors in TP53 AML. So acute myeloid leukemia is a highly aggressive blood cancer that is universally fatal without timely treatment. It's generally associated with a poor prognosis, but patients with AML that have TP53 abnormalities, either by way of deletion or mutations, have a particularly dismal outcome. So as you can see in the survival curves, those with TP53 mutations at 1 year post-diagnosis, less than a quarter of patients are alive, which is significantly inferior than those with TP53 wild type. We know that these patients respond poorly to all existing therapies, which means that we urgently need to understand biological drivers and what potentially might be Achilles' heels in this subtype of leukemia. So the TP53 abnormal AML is associated with a very unique genomic landscape. So as you can see this oncoplot of TP53 cases, there is a considerable lack of mutations that we know drive AML. In contrast, it's associated with structural chromosomal changes such as complex karyotype and monosomal karyotype. And of these, I draw your attention in particular to 5q. So deletion 5q is highly associated with TP53 abnormalities seen in about 2/3 of cases. This prompted us to ask, is there a selection pressure to lose certain gene or genes in AML with TP53 abnormalities? So when looking at this, there are approximately 40 genes within the commonly deleted region of 5q seen in AML. So we hypothesize that there are gene or genes that have a tumor suppressor function within this area in the context of AML with TP53 abnormalities. So the screen we're proposing is relatively simple because we're focused at answering a specific biological question. So we want to take human AML cell lines that are P53 wild type and P53 knockout, and then transfect these with a limited library targeting the gene specifically within the regions of 5q that is commonly lost. Our readout will be looking to see if there are particular guides that are enriched, meaning that the knockout or loss of these genes will give cells a proliferative and survival advantage. And in particular, we're interested in, are there genes that are different in a P53 wild type versus P53 mutant or lost context? And we plan to do this in 2 different cell line models to have validation. This is a really simple design experiment, and part of the purpose of this is to have a quick look to potentially inform future in vivo experiments. So other members of my lab are working on developing murine models of spontaneously mutated TP53 AML. And then if we're getting interesting results from this, we could recapitulate that in an in vivo context by seeing there are knockouts of particular genes that really accelerate disease. For this particular experiment though, because it's quite focused and limited, I'm envisaging that the cost will be under $10,000 with limited costs for the sequencing component and predominantly really in the design and construction of the library. I think we've focused our biological question to answer and help to potentially understand what it is that drives such poor prognosis in the subset of AML.
Very lovely, Yin. So questions, Dan, and then Marco. Yeah.
Maybe just to clarify, you're looking for the genes in this segment that might apply pressure that makes the cells want to lose P53. But your screen's constructed differently, right? You've got either P53 present or knockout. So could you clarify whether you're looking for a disjunction between the growth characteristics?
So we're looking to see if there's loss of particular genes in P53 lost context that will basically give the cell a survival advantage and growth advantage.
Okay, but in the people, do you think that the chromosomal abnormality comes first or the P53 happens?
It's not 100% established, but the current hypothesis is that there's actually P53 abnormalities first, usually at a low sort of variant allele frequency, and then they subsequently develop these chromosomal changes.
Great. Do you think of applying any suction pressure on this stuff? Because it might not really be enough pressure because they're transformed already, they're growing. So have you thought about and what would it be?
Yeah, absolutely. So we thought we would start this with no selection pressure, and then subsequently we can add in drug selection pressure such as chemotherapy or venetoclax-based therapy.
Okay, thank you very much, Yin. And next we'll move on to Junior.
Right. Hi, everyone. I'm Xinyu from Collab Structural Biology Division. So here I'm going to present my ideas about the pitching event. So we know in the past 20 years, we already see 3 coronavirus jumping into our human society and cause, damage like pain and panic. And it's reasonable to think that in the future, there may be another coronavirus jumping into the human society, unfortunately. And also, we haven't solved the COVID-2 yet. So here I'm just to conclude any research on coronavirus 2 that SARS-CoV-2 is still valuable and will provide valuable information towards our futures. So here I'm going to focus on the mutational effects of SARS-CoV-2 because we all know that the variants of COVID-2 come and go, and then we just sometimes not unprepared, and then we don't know what's the function and what's the effects of those variants. So here I'm going to present you a safe way to research the mutational effects of SARS-CoV-2 variants. To be more specific, I'm going to focus on the protease of SARS-CoV-2 because we know the proteases are essential for COVID-2 replication. Here is the illustration about when the COVID-2 release its genome into your cells, it gets translated into 2 polyprotein chains to maintain its replication. But this cannot happen if there's no protease activity because it relies on the protease activity to cut the unfunctional polyprotein chain to functional proteins. So we want to know what's the mutational effects from those protease on the SARS-CoV-2 replication. To examine that, I isolated the protease from the virus genome and then designed the deep neutral scanning assay to examine the activity of all the possible point mutations of this protease. Here is what the results look like. With the x-axis indicated the position number of PL-PRO's position 1 to position 350, and then with all of the mutations, 20 amino acid substitutions and premature stop codons on the y-axis here. And then on the top with the Y-type PL-PRO sequence. So the color here indicates the PL-PRO activities through our cellular assay. And then if we map this data into the PL-PRO structures, we can clearly see that so the blue means inactivation and red means the PL-PRO is active. So we can see that the residues on the PL-PRO surface is more prone to mutations, while the residues inside the PL-PRO near the core is less prone to mutations. So here we know that we can examine those activities through a high throughput assay, but the gap exists because we isolate the protease from the genome, but it's not in the virus genome anymore. And we don't know how much activity is needed for the virus to get replicated. And also, we don't know if we have a slightly inactive PL-PRO and then what's the consequence of that towards virus replication. So to address that, the ideal case is we can do this high throughput experiment in the live virus experiment, but we all know that's impractical because here we are generating new variants, and then I think nobody wants to get that done. Instead, I'm going to introduce the viral replicant assays here. So instead of having spike protein and develop membrane proteins on the genomes, I swap it to the GFP and neomycin resistant genes. So because the lack of the spike protein and those packaging materials, the genome can replicate, but it cannot be assembled and get released to infect you. So here is the result. If the genome keep replicating and then the genome get maintained inside the cells, so the GFP will be translated. And then we know that this means the replicon is getting replicated. However, if the PL-PRO or M-PRO is inactive and then makes the genome cannot be functional and cannot be replicated, then it will be degraded by the cellular machines because nobody sustains replications. So we lose the GFP signals here. So through this cell assay, we can easily tell that which PL-PRO or M-PRO, which protease variant is active in making the genome replication, and then which one is not active in not making them replicate. And we can also extend the research not only on the protease, but also we can extend it to any other proteins on the genomes to say what's the mutational effects on that. To get everything done, I will need to address the 3 burning questions. The first one is how could we maintain such a long genome through a we can't do it in traditional vectors. Second thing is how could we do mutagenesis? Because here we are talking about 6,000 protein variants or more, 200,000 DNA variants is a large number, and then to get that done, I will need to do because we already get libraries in the individual proteins. So what I need to do is just to integrate these variants into the genomes. So rather recombination methods or any other methods is quite efficient in recombination. The last thing is how could we separate the data, because we want to sequence to see which one get enriched, which one get depleted, and then the genome is so long we can't do it in bulk mRNA sequence. So instead, I'm going to perform the long read sequence like PacBio and Nanopore. So in all, the budget, which is 10K, will cover the experiment that I'm cover the basic money required to make the experiment happen. So it's like I need the money to synthesize the vectors and genomes. I need the money to do fact sorting to sort out which cell get a GFP signal, which cell didn't have GFP signal, and then I need the money to perform the long read sequence. So the outcome will be, first thing is, of course, we will uncover the residues important for protease activities. Second thing is we will identify the drug escape mutants, because with the drugs inside your systems, the drug escape one will still be active, and then the genome will still get replicated, so you can know it easily by sequencing. The last thing is the assay is not only work for COBOL2, for example, if there's COBOL3, and then we can easily apply this assay to the future researches if there's a need. And then in conclusion, this will be first introduced to WeHi, and then this is a powerful genetic screening methods, and also will help with the drug design and development. So thank you.
Questions? I'll start. I have one. So how do you make sure that you only get one virus or one back that you're assaying per cell?
So that's a good question. Everything is by mathematical calculation of chance. So theoretically, if you think that your MOI is close to 0.1, it means there are 10 cells waiting for one virus. So in most cases, it's just one virus and one cell, right? Well, any other virus didn't have the virus or resistant antibiotic resistance will be depleted during your selection.
So titration.
Yes.
Is your answer. Yep.
Yes.
Other questions? Nope. Great. Thank you, Junior. We'll move on to the next one. So,
Okay. Hi, everyone. My name is Ron Shaw. I'm from General Lab in the Immunology Division. Thank you for the opportunity to having this speech today and for the great talk and workshop earlier. So this experiment proposed is relatively simple in a mouse model compared to the other talks from the peers, which is really fantastic. So what we're trying to do is essentially trying to coupling a CRISPR-Cas9 approach to a quite unique systemic transplantation mouse model that we have established in the lab, and hopefully this can help us to uncover some novel regulators involved in T-cell transformation in thymus. Now, just to give you a little bit background to this study, I would like to introduce you of this concept of cell competition in the thymus. So for those who are not very familiar with that, thymus is the primary location of T-cell development in our body. And so it keeps pumping out mature T-cells into the bloodstream. And this process really relies on the progenitor cells coming from the bone marrow in waves. And when those cells arrive in the thymus, so the new coming cells, they can somehow outcompete the old cells that already reside in the thymus, to push them down the development pathway. And then this is really the sort of a normal T-cell turnover that we have in the thymus. And in a matter of weeks, you achieve this full replenishment of the new progenitors in the thymus. And this is quite an essential process, not only in terms of continuous producing T cells, but also it has been identified to be a tumor suppressor mechanism in the thymus. So if we experimentally block this process by introducing a wild-type thymine lobes into recipient mice without right gamma chain,
random chain knockout. So you then block the coming of the new progenitors. The old cells now residing in the thymus is known to be able to engage this aberrant self-renew program, which in a lot of times have a deadly sequence, can cause T-cell accurate lymphoblastic leukemia in mice. And this model has been reported first in 2014 by this group. And so how it looks like in the mice is that if we experimentally transplant a little stem cell lobe in the kidney capsule and the kidney capsule of the recipient mice. Now, if you do this in the wild type into wild type condition with congenital markers, and you monitor over time, what you'll be able to see is this turnover of the new cells coming in from the recipient. And in the metabolic waves, you see the whole thymus is now reconstituted with 100% of the newcomers from the recipients. Now, if we swap the rag gamma chain, swap the recipients to be rag gamma chain knockout mice, what you see is that over the time, the cells in the side, in the little lobe, will engage the self-renew program, and that in the matter of months will become a full-blown cancer and infect many different organs in the body of the mouse. And here's just giving you an example of what it's looked like for a kidney in the TIL mice. You can see that it's not all covered with these nasty cancer cells that originated from this little blob of cells. So what we wanted to do in this screening is then to introduce a CRISPR-Cas9 module into this pipeline. So what we want to do is to take a wild-type donor thymus and put them under a cultural system to specifically kill all the hematopoietic cells in the thymus lobe. So you're not left with empty thymus, but with supporting stromal cells still intact. We will then transplant engineered hematopoietic progenitor cells, being fetal liver or bone marrow engineered, into the lobe. Then we will then transplant them into the rag knockout recipient mice. From their own, we will be able to monitor the disease onset in those mice, so compare them to a wild-type setting, and also taking samples along the way in different organs, and which allow us to do the sequencing of sgRNA and to be able to see what sort of clones are turning out. And here just give you an example of what the wild-type survival curve looks like in this model. So the mice started to come down around 150 days of time. Now, what we expect to see with this screening approach is then you should be able to allow us to see the shift of the curve on hopefully both sides. So on the one side, if we have clones outgrowing with the tumor suppressor programs being impacted, then that might you end up with like more aggressive sort of cancer onset. On the other hand, if you're lucky enough, we have some recipients can hold the thymus to support the T-cell development without the disease coming up, then maybe we're hitting something really important oncogenes in that cohort. And here just a little example that we've done in this model with the B-cell knockout B-cell knockout mice that we had at Weihai, and they could act as a positive controls because as you can see how we try to knock out B-cell to family members in this model, we see a very clear acceleration in this model. And so that gives a really surprising tumor suppressor function for this for B-cell to family proteins, and we're currently investigating the mechanism underneath this. And this is just really rough cost for this experiment. So I think the biggest cost will be mouse related, costs for housing of the mouse and monitoring of the disease progression, and also some costs related with cell and organ culture, which we'll need to develop and establish in the lab. And the end product is sort of, so on the one side, it might give us a list of novel tumor suppressor genes, for example, SBCL2, but not necessarily acting in the same way. And this could give us implications in other sort of types of cancers with the stem-like population, as we see here, which can engage the self-renew program. And on the other side, maybe we can also identify some important oncogenes. And that's basically the end of this talk.
I think Marco.
Very nice. What's the library you use? Focus?
Yeah. That's a great question. So because you can only introduce a quite limited number of cells back into the Science log, so we are thinking more like a targeted, a small library, either targeting a T cell development, sort of related pathways or other like known here oncogenic sort of pathways. Yeah, that's the plan for now, I think. Yeah.
I have a follow-on question. How do you deal with the potential confounder that those factors may also have a role in the hematopoietic stem cells or the hematopoietic stem and progenitor cells that you'll need to transduce in the first place?
Yeah. So you're thinking about hitting something too essential, so they won't support the graft to have a normal.
Yeah, or maybe just differentiating between the role they might have in the HSPC that you do the transduction in, and then the readout, which is once you put them into a virus.
Yeah.
They're kind of two different. They're going from the stem to more differentiated. So they
yes.
could have a function anywhere along.
Yeah. Well, it's still kind of a black box situation, I guess. And I guess the results can give you some idea. Like if you hit something really different, you can still have like a result. But then how to interpret back to your initial question would be like carefully considered, I guess. And then, but yeah.
All right. Thank you. Thanks very much, Manjo. And last one. Ollie's talking. It's on here. Yes.
As well, because
yes.
they may want to answer
So
some
we
questions.
have a trio. Like one's going to pick and the others might answer questions. So yeah, come on.
Yeah, I just have a very talented second year undergrad here, so she's part of the team as well. Okay. Yes. So I'm just going to begin, and first I just want to say I'm very privileged to be here and to be able to present my first pitch ever to you guys. So it's really exciting alongside my teammates Laura and Olivia. So today I'm going to be talking about genomic dark matter, and a lot of you are probably wondering, what do I mean by that? So with traditional Cas9 genome-wide knockout screens, we primarily only target protein coding genes, and that's somewhat of a problem because the large majority of the genome actually remains untapped, as you can see on this diagram on the right, in which only 1.5% of the genome is actually contributed by protein coding genes, which is a huge problem. And a prime example of these are long non-coding RNAs, which are around 200 nucleotides or longer. And these play important roles in cancer, often deregulated, and can also be targeted as biomarkers. So how would we go about targeting these using a screening method? So now I want you to imagine if we had a CRISPR screen that was capable of targeting both protein coding and non-coding transcripts, such as LNC RNAs alike. And it had the capability of being inducible, rapid, and reversible for transcript-specific depletion in mammalian cells. So how exactly would we go about doing this? Well, I'm going to introduce to you a rapid, reversible, and inducible system. In this system, we begin with a high-fidelity version of Cas13d. If you're not familiar with Cas13d, it's an RNA targeting Cas, and as such, it has a couple advantages over Cas9. One of which it's more rapid and more targeted because you're actually targeting the transcriptome, not the genome. And furthermore, you can actually isolate specific RNA isoforms, which is a key advantage of this technology over Cas9 screens. Furthermore, we would complex this with a Degron tag so that we can acutely deplete the endonuclease itself by recruiting an E3 ubiquitin ligase complex. And then that would obviously recruit the proteasome, which would degrade the protein of interest, in this case, the endonuclease. And that has a lot of advantages because we can actually reverse any transcriptome-wide changes. And then we can start to look at phenotypes, not only when we knock down the RNA, but also when we allow it to replenish, which has a lot of interesting insights, not just for our screen. And then furthermore, we complex this with a DOX-inducible guide vector. And by doing so, we would have temporal control of transcriptome by editing. But it doesn't just end here. You see, this type of technology has lots of different utilities, one of which is it's scalable to single-celled systems. And furthermore, these types of libraries targeting non-coding RNAs, and they could even be designed in kind of like in combination with protein coding libraries to be used throughout the whole institute for a variety of reasons, not just for our lab in particular. So here we are asking for approximately 10,000 Australian dollars to achieve this. You can see kind of like the costs delineated there on the right. But ultimately, even though we have a conservative estimate of $8,800, we know that not everything goes as planned in science. So I think the $1,000 buffer is probably a good idea. So now, just to summarize, we present to you a transcriptome wide screening method that is both rapid, inducible, and reversible. And by performing these types of screens, we can target non-coding transcripts, which can broaden our scope for new therapeutic targets for cancer, but also for many other diseases. Yeah, that's pretty much it. So thank you. You want to come up?
Thank you, Ollie and Olivia and Laura. So, yes.
Really interesting technology. I was just wondering, do you already have a plan for context or assisted image detection?
Yeah, so I just completed my first genome-wide CRISPR-Cas9 screen, and ultimately, kind of leading on to the question that I was asking Marco, we were thinking of using a different modality to kind of verify our findings. So obviously, we could just use a competition assay, or we could design a more targeted library. But in addition to that, we could also use our library that we design as part of this project, kind of as a proof of concept as part of that project, just to show that it works. And then from there, we can always build library size. We can start to look at other mechanisms. Our lab's involved in transcriptional regulation, so I'm looking at the elongation phase of transcription in particular, but there are many other use cases. So yeah.
Okay.
Very nice talk. How many of the mRNAs or RNAs in general are remaining in the nucleus and never come out? And does your Cas13 cells as well?
So yeah, so we actually can complex the Cas13 constructs with nuclear localization signals, and they have been shown to target nuclear transcripts. So I don't know if that answers your question. Yeah, yeah.
Yep. Yep.
Yeah, so the Cas13 family of enzymes is always kind of leading actually. I'm not sure that you guys now have done the experiments.
So that's actually true. Like traditionally, Cas-Rx, which is like kind of like the gold standard of RNA targeting Cas, does have reported off-target activity and collateral activity. But more recent, there are some preprints looking at DJ Cas13D, which is an ortholog of Cas13, alongside a high fidelity version of Cas13D, which kind of fixes those problems with the previous generation. And also, I know I haven't discussed this in my talk, but there are many other ways we could do these types of screens, right? And you may be thinking we could use like short hairpin RNAs and things like that. But ultimately, they too have off-target effects and a whole variety of other disadvantages, which I won't go into right now, making them not as suitable as what we have presented today. Yeah.
Last one, Andrew. Yeah, last one.
Where in the cell does Cas13 target RNA?
Yeah, so it can target transcripts like.
Sorry, I think, yeah, you probably didn't.
Yeah, we just answered that.
Yeah, I should have done the repeating the question.
Sorry, yeah, no, it's fine.
Right, we're going to leave it there. Thank you to all the students for fabulous, wonderful pictures. They were really, really good. And so we're going to convene, but there's afternoon tea outside, and there's a barista there, so it's barista coffee. So just order yourself a coffee. If it's not too late in the day for coffee for you, or some other hot beverage, and we'll be out soon. Thank you very much, everyone.